All right, class. So let's start by looking at electrical synapse first, then we will move on to chemical synapse. So this diagram on the left hand side is showing us an electrical synapse, which we find in the smooth muscle cells, cardiac cells, and also in the central nervous system. Now, the little box on the extreme left right here is showing you the cardiac tissue. If you zoom in, if you blow up the intercalated disc of which kind of joins the cardiocytes together, this is what it looks like. You have, this is one cell and this is another cell, and the two cells are connected to each other by these channels, and these are called gap junctions. Now the gap junctions help connect the cells together, and through these gap junctions there will be flow of ions from one cell to another, and this is how electrical synapse works. All right. Now, looking at chemical synapse, in case of a chemical synapse, there is always a neurotransmitter involved. And in case of chemical synapse, the upstream neuron and the downstream cell, they are not touching each other. They are in very close proximity, and the area in between, there is a small area in between, and that's called the synaptic cleft. We also have talked about a presynaptic and a postsynaptic cell, and know that these two cells are somewhat held in place by some adhesion molecules, also known as CAMs. Okay, so let's draw out a synapse. So here we have a presynaptic neuron and a postsynaptic neuron, and this area in between is called synaptic cleft. I want you to understand that the pre and the postsynaptic neurons are not physically touching each other. There is space left in between, and that is called the synaptic cleft, and always there will be a neurotransmitter involved. So let's take a look at a synapse between neurons. So we have a presynaptic neuron and a postsynaptic neuron. Okay, now I will blow up the synapse area. Let me see if I can change color. No, I cannot. I'll blow up the synapse area, and we will see what exactly happens in this area. Okay. Okay, so right here we see the axon terminal. We see the axon terminal of a presynaptic neuron. And we see the postsynaptic neuron, right? So what's happening here? Now, an action potential will travel down the axon of the presynaptic neuron. So we have the action potential here. And as the action potential reaches the axon terminal, remember, voltage-gated calcium channels open up. So these channels that I am drawing, these are voltage-gated calcium channels. Now these channels open up. We also know that we have a lot of calcium outside the cell as compared to inside. So when these channels open up, obviously calcium will flow inside or flow into the cell. Calcium, so now we have calcium inside. Now calcium causes these vesicles. What are these vesicles called? If you remember from muscle lecture, these vesicles are called synaptic vesicles. And these vesicles are filled with neurotransmitter. Okay, so calcium will cause these synaptic vesicles to fuse with the presynaptic membrane, and exocytose acetylcholine, not acetylcholine, in this case actually we are talking about any neurotransmitter. And as the synaptic vesicles are fusing with the presynaptic membrane, the neurotransmitter is released into the synaptic cleft. Okay? I need to change color. That's why I minimized it. Okay. So one more time. We do see the axon terminal of a presynaptic neuron, and we also see a postsynaptic neuron. As action potential reaches the axon terminal of a presynaptic neuron, voltage-gated calcium channels open up and calcium rushes into the cell. Calcium causes the presynaptic vesicles to fuse with the presynaptic membrane and exocytose neurotransmitter in the synaptic cleft. So now we have these neurotransmitter floating in the synaptic cleft. Also remember, we have a lot of sodium in the synaptic, outside the cell. So outside the cell we have lots and lots and lots of sodium. Okay, we have low sodium inside. We know this. Okay, all right. Now what will this neurotransmitter do? Well, there are ligand-gated channels on the soma in this case. So these channels, the orange channels that I'm drawing, these are ligand-gated channels. Ligand means a chemical, and the neurotransmitter will work as that chemical. And the neurotransmitter will bind to these ligand-gated channels okay, and as the neurotransmitter binds to these ligand-gated channels, they open up. We talked about this before. And as they open up, sodium will enter the cell. What will sodium do? Sodium will depolarize the cell because it's positive charge, and sodium will depolarize the postsynaptic neuron, and if the depolarization is strong enough at the axon hillock, an action potential will be generated. Okay? Now, is it always that sodium enters? Or can we have different neurotransmitters opening up different gates and letting in different types of ions? Well, let's take a look. I will start, actually, I'll draw another one. Okay. So, let's get started. We see axon terminal of a presynaptic neuron, and this is a postsynaptic neuron. You need to listen to me very, very carefully. So in this case, we have an action potential that reaches the axon terminal, which causes these synaptic vesicles filled with neurotransmitter to fuse with the presynaptic membrane and exocytose the neurotransmitter in the synaptic cleft. Okay, now listen carefully. These neurotransmitters are ligands, right? And the soma has different channels, many different channels. And I will draw multiple channels here. Okay, now, depending on what neurotransmitter is being released by the presynaptic neuron, we can have ligand-gated sodium channels open up, and in that case, sodium will enter the cell. Sodium brings in positive charge, so it will depolarize the downstream neuron. It can also open up potassium channels, and in that case, potassium will enter the cell. It can also open up potassium channels, and in that case, potassium will leave the cell, because potassium, remember, we have more potassium inside the cell as compared to outside. Depending on what neurotransmitter we are looking at, it can also open up chloride channels, and in that case, chloride ions will enter the cell. Chloride brings in negative charge, so that will hyperpolarize the downstream neuron. It can also open up calcium channels, ligand-gated calcium channels, and calcium will enter the cell. As calcium enters the cell, we will have depolarization. So let's play this again. So neurotransmitter. So depending on what neurotransmitter, let's say we have neurotransmitter A. If the neurotransmitter A can open up ligand-gated sodium channels. If ligand-gated sodium channels open, then there will be entry of positive charge that will cause the postsynaptic neuron to depolarize. Okay, next, let's say we have another neurotransmitter, B. And this neurotransmitter causes ligand-gated potassium channels to open up. And when these open up, we have potassium going out of the cell. So we have exit of positive charge that cause hyperpolarization of the cell. We can have another neurotransmitter C that might cause the ligand-gated calcium channels to open, and when that happens, we have entry of calcium into the cell, meaning we have positive charge coming in, and that will cause the cell to depolarize. We can also have D. We can also have ligand-gated chloride channels, and when these channels open, we will have chloride coming in, entry of chloride, and if we have negative charge coming in, it will also be hyperpolarized. Does that make sense? Please go over this again and again and again. And I also want you to remember the 4 ions I mentioned you are responsible for: sodium, potassium, calcium, and chloride. You must know which is in high concentration inside the cell, which is in high concentration outside the cell. To review, we always have high potassium inside, and the rest, sodium, calcium, and chloride, they're always high outside as compared to inside. Okay? So I might ask you, like, okay, so let's say we have a neurotransmitter, and due to this neurotransmitter we have ligand-gated potassium channels opening up. Which way will potassium move, and what will happen to the downstream neuron, or what will happen to the postsynaptic cell? So I'll start with a new video.